Okay, we're back for part two. We'll look at a little bit more detail about the information we can get to tell us about the quality of calibration lines. Calibration is important, so we'll talk about this. We'll talk about the process and a little bit about planning experiments. I'll look at some examples and some further resources in the tutorial activity. Then we'll think about how we can answer some of the questions which this particular lecture is going to raise. Okay, calibration. Results are meaningless without calibration. They're just a number floating around in the infinity of numbers that exist out there. We need to have something to tie them to. And we typically do this by using standards from known concentration. I've done this quite a lot. We did this in the first year for various experiments. We create a set of standards of known concentration. We measure a particular instrumental response. It might be by refractive index, might be using HPLC, infrared UV detector. We plot a calibration line. We get the linear regression statistics, which is the equation of the line and perhaps the R-squared value. And then we measure the instrument response for any samples and use the calibration line to calculate the concentration of the analyte of interest in the sample. If you remember, anything you see in this context, which has a square bracket around it, it means concentration of. So in this case, concentration of sample. Okay, it's a calibration process. Plan the experiment. Now, in most cases, of course, you get the experiments method given to you. But if you're doing this as a final year project, it may be that you are doing a project on method development. So next slide will probably be a particular interest to you. We take the measurements, we apply the calibration line, we get the regression statistics, we determine the concentration of the sample, and then we do some estimation of the uncertainty of the sample results, which we're going to talk about a little bit more here. Yeah, so if you're planning an experiment, there's a number of things you'll have to do. You don't know what your concentration range for your standards are. You might say, again, we've got some orange juice which contains vitamin C at a particular concentration, therefore we need our standards to be within that range. That may mean we need to dilute the sample. There's a number of things we might have to do. Decide how many samples we need within that range. Typically we do 4 or 5, and we do that for convenience. Obviously do 9 or 10, it's better. You get better results, but it is time consuming, and in most cases we'd often get away with doing 4 or 5. Decide how many replicates you need for each standard. There's a clue at the bottom of the page. Prepare the standards, decide how many replicates you need for each sample, and again the clue is there, triplicate is good, and we carry out the measurements and then do our downstream analysis. Okay, here's some typical results from HPLC for beta carotene, I think. This is for a standard. We're saying after 15.9 minutes the beta carotene standard comes out and has a peak area of this for a particular concentration. I don't know what the concentration is, but that's what we would plot. For the sample, it's much more complicated. We've still got a peak at 15.963, which is our beta carotene standard, our beta carotene sample measurement rather. And again, we can get the peak area there and plug it into our calculations. Okay, take a step back. Go into the importance of calibration. Say it's 4 peaks for the HPLC of sugars and orange juice. We just taken some orange juice, filtered it and put it down the HPLC and got 4 peaks back. But what do they mean? There's a number of things we need to know. We need to know what the actual sugars they are, a qualitative measurement, and how much there are of each, which is a quantitative measurement. Okay. So to do the qualitative thing, with qualitative estimations, we use standards. We have a standard ever sucrose, and we say, well, it comes out at about 7 minutes. So it's reasonable to assume that in our sample, the peak at 7 minutes is sucrose. And doing that, we can then, that's sucrose. We can then come up with an estimate of at least some of the peaks there. So we've got standards of sucrose, fructose, and glucose. There's peak 1, which we don't know what it is. Maybe sugar, may not be sugar, but we don't have a standard for it, and we're probably not interested in it. Okay, so that tells us what will come out at what time under the conditions running this particular HPLC experiment. We then produce a standard line, as we discussed already. Create a set of standards of increasing concentration, measure the peak areas and plot them against concentration, where we've seen many times before. So there's an example one for sucrose in the analysis of orange juice. We've got sucrose up the y-axis with the millimoles. Remember, we must always include units where they exist, which is most cases. and the peak area in millivolts, the detector response. Now back in the day, what we'd have done this on graph paper. We'd have drawn a line across and dropped a perpendicular down to the peak area and then worked it out on that basis. These days, of course, we would use Excel. Yeah, okay, so dropping a perpendicular. And there's our sample. These days, of course, we just put the results into Excel and get the straight line, put the trend line through it, and get the straight line data, which will allow us to rearrange to solve the concentration of X, the concentration of the sample. Now we've covered this. There's a number of resources here. There's SWAY there, which we did in first year on straight line graphs. And there's a couple of videos there, one on converting HPLC results into Excel, another one on standard deviations and error bars. So you probably want to revise these. There is a SWAY associated with these lectures, as you know, and you can access these videos directly from there, or from this presentation. Okay, a couple of tutorial activities. In that Excel spreadsheet, which is referenced at the bottom of the slide. In this case, you basically got some data, and you need to calculate the average peak area for the samples, get a standard deviation, plot a calibration line, obtain a trend line, get the equation of a line in the R-squared value, and then rearrange to solve for X. It's also asking you, using the standard deviation, to add error bars. Again, see the video. Make sure you add all relevant titles, labels, units to the graph, and then, as I mentioned, calculate the concentration of beta carotene. And you get a graph with something like that. Okay, quite a nice graph. Okay, so we've got some outstanding issues which we'll look at in the third part of this lecture. How low can we go? What concentration can we measure down to? Is R-squared telling us all we need to know? And look at the important factors, the limits of detection and limits of quantification, both of which are derived from something called residuals. It's a slightly more sophisticated statistical way of looking at our results. Okay, that's all. I'll see you in a bit.